This is episode 307 of the Stem Cell Podcast, Vascular Biology, with Dr. Juan Melero-Martin. Hello, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. This podcast is brought to you by STEMCELL Technologies, a global biotech company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, please rate us, leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, as our guest, we have Dr. Juan Melero-Martin, who's from Boston Children's Hospital and Harvard Med School. He's on the podcast to talk about his research on vascular network formation. We've also got our usual roundup of recent highlights in stem cell news. That's coming right up.
But first, we'd like to remind our listeners about ESC and iPSC news, one of Stem Cell's free bi-weekly scientific newsletters. ESC and iPSC News summarizes all of the latest research, news, jobs, and events in ESC and iPSC research and delivers it right to your inbox every other Wednesday. Save time and keep current with ESC and iPSC News. Subscribe for free at www.escellnews.com. We'll start things off with a paper coming from a former guest of the show. That's Dr. Deepak Srivastava, somebody who I've known for many years as one of the icons in cardiovascular biology and stem cell biology as well. And of course, he's well known for his involvement with the ISSCR. Claim to fame back in the day was direct reprogramming. And of course, they've, focused on congenital heart disease and developmental biology over there at the Gladstone for years now. And in addition to his partner in crime, Benoit Bruneau, of course, we also have had on the show. The Gladstone is just an amazing place for studying developmental biology and connecting it to the clinical applications. And that's really exactly what they're doing here in this Nature paper that just came out from Deepak Srivastava's lab. First author here is actually somebody who I know pretty well, this Sanjeev Ranade, who's a junior investigator who just started his own lab down in San Diego. So shout out to you, Sanjeev. Title of this paper is Myocardial Reprogramming by HMGN1 Underlies Heart Defects in Trisomy 21. And, knowing Sanjeev, I've known about this story for many years now. It's been more than half a decade in the works. And the focus here is on trisomy 21, of course, commonly known as Down syndrome. And associated with Down syndrome, trisomy 21, are cardiac defects, and congenital heart defects are, as we know, the most common developmental abnormalities, unfortunately affecting around 1% of live births. And over the years, there have been an evolution in the model systems to study congenital heart defects. Like, for example, we talk about cardiac organoids most recently in our field, in the stem cell field, and something my lab is actually focusing on as well. Really exciting, scalable, high throughput model systems to study the basic mechanisms of how these congenital heart defects arise. But this is actually kind of an old school paper focusing more on the mouse models, less so on the stem cell models. And so diving into it a little bit more, we know that aneuploidy actually causes around 15% of all congenital heart defects, with trisomy 21 specifically being actually the most frequent form. And these congenital heart defects, or CHDs, occur in about 50% of cases of Down syndrome, with around 1,000-fold enrichment of atrioventricular canal, or AVC, AV canal defects, that can mess up the junction between the atria and the ventricles. And of course, this is really critically important for proper blood flow, proper oxygenation in the early developing heart. And a lot of times, if the defects are severe enough, upon birth, these kids immediately have to have, cardiac surgery, pretty rough stuff. So the AVC, atrioventricular canal, contains different unique myocardial cells that are essential for the development of the valve and development of the cardiac septum as well. But the connection between Downs and the AVC defects, it's not really well understood. In particular, the combination of different dosage sensitive genes on chromosome 21 that are responsible for these, Down syndrome associated CHDs are not really well known. So you have that extra copy, right, of chromosome 21. And the question is, what does that do to altering cardiac development, right? And it's amazing that that connection hasn't been fleshed out for many years now. And we like to hate sometimes, I don't know, hate is a strong word, but we focus in this age of NAMs more on these alternative model systems other than animal models. But I would say the mouse models and animal models have been extremely important for understanding cardiac development specifically. Um, Even though, of course, there are physiological differences between mouse hearts and human hearts, certainly the mechanisms in part are conserved. So this is a situation where the mouse model and the rodent systems have been really useful. So they did use pluripotent stem cells to some extent here in this study, but they also use mouse models of Down syndrome and actually identified HMGN1, which is specifically a nucleosome binding epigenetic regulator encoded on chromosome 21 as a key contributor to these atrioventricular canal defects. And they did what Deepak's lab is really good at, a lot of single cell transcriptomics, epigenomic analysis, showing that the trisomy 21 in the situation of Down syndrome can actually shift the human atrioventricular canal cardiomyocytes towards a ventricular myocyte state. So like a shift in the cell lineage there, which is really interesting. And then they did a little bit of crop-seq, CRISPR activation, single cell RNA droplet seq, all the rage these days, of different chromosome 21 genes that are expressed during heart development, and actually showed that, an upregulation of HMG and 21 can mimic that shift to the ventricular myocyte state. So it's a really cool story looking at developmental shifting of trajectory, specifically in myocyte specification, atrial versus ventricular, right? But on the other hand, if you actually delete one of the HMGN1 alleles in those trisomic cells, you restore the proper gene expression, as you might expect. So as opposed to 3 copies, you shift it back to 2 copies and you get proper gene expression, and you actually get a restoration of the myocyte fate as well. So in the mouse model of the trisomy 21, you had the similar transcriptional shift of the AVC atrioventricular canal myocytes that was restored by a reduction in HMGN1 gene expression, right? And the phenotype is the most important thing here. That correction actually led to a rescue of those valvuloseptal defects. That's pretty incredible and really striking finding. So we always talk about here on the show about certain genes that can serve as nodes to driving some of these critical phenotypes. And in the best case scenario, you have a single gene node like HMGN1 here as a dosage sensitive modulator. That's what they call it, of atrioventricular canal development, cardiac septation in the context of Down syndrome. And so I think it's an amazing study from the basic science perspective, because you're really getting to the bottom of what is it about in the situation of trisomy 21, what actually leads to those defects in the developing heart. And, I don't think there are direct therapeutic applications to this, unless you can, I don't know, somehow very early reduce the expression of HMGN21 in utero or something. I don't think that's the angle they're going for. But I think more importantly, it's a way they offer a paradigm to figure out these aneuploidy associated pathogeneses, right? Using these in vitro and in vivo systems to actually map the different genes that are causing these, complex genetic disorders like trisomy 21. So I'm really excited about this because it's a beautiful basic science cardiac study with, of course, some pluripotent stem cell elements in there as well. But It's getting to the bottom of the question of why are these kids with Down syndrome born with these cardiac defects? And it might honestly just come down to the dosage of this single epigenetic regulator in HMGN21. Just a beautiful study here.
Yeah, this is a great mechanistic study. It's what they do over there. Basic developmental biology, deep mechanistic insight. And they're unleashed to do that. And I think that maybe in part there's a little bit of a wrapping here of like the clinical implications. They kind of lead with this idea of heart defects and trisomy 21 Downs as like a clinical target, which I think is I don't want to say it's misleading, but it's kind of like not the point at all, as you just kind of stated, alluded to at least. It's not really a clinical target here that we're identifying, but more of a well, first, like the mechanistic detail here and the model and approach and the tech, as you said, the crop seek, and then going back into a mouse model where you have the real developmental physiology that you can't get in non-animal models. Let's be honest about that, particularly in the case of the heart. But more than all that, it's you said there, the framework, right? It's a conceptual advance, um, which I think now enables you to think differently, both in terms of like the clinical diagnostic, but also the approach, perhaps therapeutic, to congenital heart defects, which are super common. But the etiology and the range is so broad that it's hard to understand, probably polygenic, and probably owing to, in part, just the complexity of this bioengineering marvel that is the heart, particularly in development. So I think it gives you a different framework for understanding. I don't know that I would love to have seen, but is a Nature paper. Um, I'm waiting to see, I guess, building on this, how this relates to that other, what, 85% of heart defects that you find outside of the context of trisomy, the more common, maybe spontaneous or aneuploidy based defects. So a lot of foundation built here, and I expect to see a big impact moving forward.
Yeah, absolutely. And I like what you alluded to, the downstream application to other genetic disorders of the heart. And they didn't really focus on organoids as much here, but if the organoid models are getting advanced enough, ips derived organoid models, and scalable and high throughput enough, and with vascularization and all the things we talk about, then certainly you can incorporate those into a study like this. They think they would fit really perfectly, make iPSCs from folks with Down syndrome and see how the cardiac organoids that are developed are altered in the trajectory of their development. So I think, it's an exciting time for these NAMs, next generation model systems, that the NIH is really emphasizing these days. But again, this is also a reflection on the power of these in vivo systems, especially for cardiac development and really teasing apart some of these fine mechanisms.
Yeah, and a story about cell fate, really fundamentally, and divergent cell fate, kind of uh, pathological cell fate, and we're going to come back to that in this roundup, kind of the theme. But first, I have a different kind of segue from this, which is leaning into this aneuploidy angle. So going back to early, the earliest points of development, aneuploidy in pre-implantation stage embryos, really a major clinical target. Pre-implantation genetic testing is something that's becoming more and more common, obviously in the context of IVF. But we're getting to a point of, as they call it, PGT all, where every embryo is being tested. But when you look at human embryos, pre-implantation stage, particularly blastocysts, you see a mix of both normal euploid as well as aneuploid cells. And of course, there's like aneuploidies of meiotic origin in some cases, which are going to affect all of the daughter cells, so all the cells within the embryo. But what you see most commonly is mosaic aneuploidy, which is due to mitotic errors after fertilization, right? Hence the mosaic element. And those can contribute, those aneuploidies can contribute to implantation failure and embryonic arrest, sorry. But it's also thought that aneuploidy it can be tolerated, particularly if that aneuploidy is seated in the trophectoderm cells that are ultimately fated to form the placenta, right? And when you look at the numbers, it's actually pretty startling. Mosaicism, you'll see mosaicism in roughly half, as many as three-quarters of embryos at cleavage stages, and approximately a quarter of blastocyst stages. So, like, you know, a lot. Um, and uh, there's been a lot of study of the mitotic errors in cleavage stage embryos. But as the cells get more multiple and the metabolic demands of the embryo increase, it's harder to track this. So there's not a lot of studies of embryos and mitotic errors emerging in later stage, approaching blastocyst embryos. And part of that's due to the approach. So microinjection is one way you do it. You inject some kind of cell intrinsic marker that'll tag the nuclei, but it doesn't really work for most embryos donated for research because they're obtained later in development when there's already tens, if not hundreds, of cells. And often blastocyst stages are they're already at blast stage, and live DNA dyes have been shown to negatively impact mitotic progression and damage DNA. Add to that the kind of optical method, so confocal can be really phototoxic. So all these forces converge on not a lot of data, particularly in human embryos looking at um, around blastocyst stage development. So enter light sheet microscopy. All right, and this is a study from led by Kathy Niakin's group at University of Cambridge at the Francis Crick Institute. And what they did is they used light sheet and they systematically tested all kinds of different nuclear labeling methods, ultimately settling on this electroporation method with DNA tagging to introduce mRNA into human embryos at the blastocyst stage. And they developed this open source semi-automated segmentation method, which was like deep learning, all kinds of machine learning, I don't know, tech, software algorithms, in order to track these cells. And what they found is really important. I would say the clinical implications are, I think, vast, but also just fascinating from a developmental the biology standpoint. What they found is that in some cases there were micronuclei, these little offshoots of DNA that were passively inherited by a daughter cell, right? And in some of those cases, they would not be reincorporated into subsequent mitoses, so they just float around as like little blebs and contribute not at all to the genome, so no risk there. But in other cases, you would get incorporation of these micronuclei, right? So a mixed bag, but certainly some cases where you would risk the aneuploidy. The important thing is that in most of the cases, so the vast majority of cases where you got that reincorporation, those cells remained externally positioned, right, in the trophectoderm layer, which is consistent with contribution to the placenta as opposed to the inner cell mass. And this is something that we covered before on a roundup, probably more than a year ago, where we noted it was observed that the trophectoderm was much more prone to mitotic errors and aneuploidy than the inner cell mass, which was comforting news. The really important implications of this is that we do a ton of PGT, right? Pre-implantation genetic testing in the context of IVF. And what are we biopsying? The troph, right? We're biopsying these trophectoderm cells, which we already knew have an increased likelihood But here we're showing definitively that though there is a enriched or overrepresentation of aneuploidy relative to the inner cell mass. So, like, it's a false positive, effectively, of aneuploidy in many of these cases, which also jibes with clinical observations that transfer of mosaic embryos often leads to healthy conceptus live-born uh, uh, babies that are healthy, right? So this is a big deal. One, because of the tech and the convergence of all this tech for live imaging, which we're going to come back to a little bit later in my next story. Um, but also here, I think we got to really revisit the rationale for discarding mosaic embryos, and what, like, I don't know if the number needed to treat, or the point being the specificity of our biopsy results in the context of IVF, especially in an era of PGT all.
Yeah, I think a lot to tease apart here. And for me, as somebody who's not directly involved in this field like you are, I was uh, amazed by the technological innovation that's leading to, these findings. And part of me is like, these are such critically important results that they're obtaining here. um, is this something that, and I'll ask you as the resident expert in this topic, is this something that we would want to incorporate into standard, IVF workflows, that sort of thing? This detailed level of tracking and analysis and AI driven. And I know, I'm sure there's folks already starting to do this kind of stuff, but what is the practical incorporation of some of these workflows into IVF?
Absent uh, a, like, clinical indication where we're moving toward, I guess, genetically repairing affected embryos, I don't think that we want to be doing any kind of electroporation of exogenous genetic material into embryos, just because of the unknown unknowns there. So I don't think this is something that is going to become uh, incorporated uh, at all, really. Also, just because of these days we have more information, right? Than we can really use, and oftentimes it leads to unnecessary intervention. I think at this point, this would definitely qualify as over data, uh, but the potential for study and the potential for analyzing patients who have a history of embryo arrest, or you know pregnancy loss, early pregnancy loss, or failure implantation. I think that's where this will become one of the major arrows in the quiver. Is you'll have a patient who's having a lot of challenges, and they'll go to, let's look at a live analysis. Let's do mitotic arrest analysis. And I think that could be a convention in IVF that would be really, really useful for those 5 to 10% of patients that just repeatedly fail.
Wow, 5 to 10%. I didn't realize that was such a high number. Yeah, that's incredible. And yeah, hopefully this technology can enable some hope for those individuals. So staying in the realm of reproductive biology, I'm going to shift to a cell stem cell paper that just came out that's um, on the topic of women's health as well, women's reproduction, reproductive health, and this is on looking at the impact of vitamin C. Of course, vitamin C is in orange juice, all the great juices, right? Apparently has a protective effect on primate ovaries. We're talking about ovarian health here, ultimately leading to proper generation of follicles and oocytes and so on. And they did this predominantly in a primate model, so non-human primate model. This is coming from Guanghui Liu over at the State Key Laboratory in the Chinese Academy of Sciences in Beijing. First author here is Ying Jing. So we know ovarian aging is important. Obviously, it plays a really critical role in female reproductive health with different implications for treatment strategies, quality of life, right? So on. And, of course, the population as a whole is undergoing significant aging these days, and average age of, of reproduction, and especially in certain subpopulations, certain countries is definitely increasing. So these are all things that we have to consider when it comes to aging and women's reproductive health, right? But the potential of one single agent, right? Like everybody kind of looks for that magic bullet, right? What's that one magic drug, agent, whatever, that may be able to counteract ovarian aging in this example? And sometimes, most of the times, there's no such thing as a magic bullet, right? It's way more complex than that. Um, but at least in the context of this specific study, they seem to have found something that may somewhat reverse that concept of ovarian aging. And they definitely have to take a deeper dive into this, but it was pretty well done. It was a more than 3-year study um, in monkeys that was demonstrating that oral vitamin C can actually have geroprotective effects. So aging protective effects against the aging of the ovaries in these female monkeys, for example. And why does it do this? How does vitamin C, actually have this mechanism of geroprotection? It diminishes different aging biomarkers, reduces oxidative stress, reduces follicular depletion. And then they took a single cell dive into it, as we always do, and had a single cell transcriptomic clock, that's what they call it, and showed that vitamin C specifically can reduce the biological age of oocytes by more than a year, and of somatic cells, in addition to the oocytes, by more than 5 years, 5.5 years. And again, they're using different biomarkers to actually, you know, characterize the biological age of the cells in these monkeys, in these primates. Um, and they took an even deeper dive mechanistically into it and figured out that the effect is partially mediated by the NRF2 pathway, which is alleviating the senescence of different ovarian cells and also reducing inflammation. So, it's building on something that we already kind of knew about the importance of vitamin C just across the board, not just in the context of ovarian aging, but just in overall health in mammals and primates, right? We all got to take our vitamin C, of course, right? But specifically, the power of this study is figuring out the role of vitamin C in combating primate ovarian aging, and maybe even some insights for developing different interventions against human ovarian aging, as simple as drinking a tall glass of orange juice every single day. I don't know. That's a stretch, of course, but that's the concept, right? We're all very interested as our populations age. We're all interested in these strategies to counteract those aging effects, not just in the somatic system and in the somatic tissues, but also in the reproductive tissues as well, and in the ovaries. You can perhaps find equivalents for this in male reproductive biology as well. So I don't want to say this is a magic bullet because nothing is really ever that simple, right? But perhaps it's something to take a deeper dive into the idea of using vitamin C perhaps as a geroprotective agent.
Yeah, I'm very impressed by this because it's what you said. It's one of those things that you knew that was so taken for granted that like you almost thought it was overstated or almost was like so true was untrue. You know what I mean? Like, oh yeah, scurvy. Like vitamin C, if you don't have it, like yeah, you'll get sick. But like, is there really a health benefit by having, more than you fundamentally need? Um, and of course, I mean, why am I so surprised? Because I'm unimaginative, I guess. The it's an antioxidant, right? And as you said, the whole idea of vitamin C being implicit for just general health and here being applied for reproductive health, I'm kind of thinking it in reverse because, clinically, realistically speaking, the problem here with the problem is that the patients come in and they're already infertile, right? And or they're already struggling with fertility. So I don't know that vitamin C here would be like, you know, uh, you know, rejuvenative. I don't know that it would rescue anything. So I'm thinking more just in terms of my general health because and everybody's general health because, the reproductive system is a nice little laboratory within the body where all these deficits become manifest, maybe or overt in advance of when the same type of fibrosis, oxidative stress, whatever it is, immune infiltration, sequelae thereof, like, all those things become manifest in the ovary in particular because it's such a dynamic organ undergoing all this, cellular dynamic, motility, et cetera, et cetera. So, I'm thinking if vitamin C works for the ovary, then it probably works on some level everywhere, and I'm gonna start drinking my OJ and more forget about me, I'm doomed. But the kiddos, I was resistant. Juice in general, Arun, is kinda like we're a big campaign against juice because of the sugar, and I get that, and I think that's real. But I wonder if, like, a serving of orange juice a day, why not? If you keep it there, could be good.
Oh, for sure. Yeah. I mean, I'm not anti-juice, but not the biggest sugar guy, but just eat an orange, right? Something as simple as that.
Yeah, to get the fiber too, you're right, that's even better.
Yeah, get back to the source, you know? Yeah, I think it's this study is reflecting on something in general that we already knew, the importance of your vitamins, importance of taking vitamin C and all these kind of things. And I like what you said, how reproductive health is kind of a window into overall health, right? And you see that perhaps negative situations of reproductive health are just a reflection on negative situations of the overall body, right? So um, I like that analogy, and there are things to, of course, mention in this study. It's certainly exciting, and anytime you use a non-human primate to make these kind of observations and conclusions, and it's we've acknowledged that this is very expensive, very hard to do, and I know certain areas and institutes in China actually do this very well. But there's still a really small sample size here. One thing that's important is if you're talking about reproductive biology, you want to look at those functional reproductive outcomes, right? In terms of the ability of these monkeys to actually have babies and all that kind of stuff. And they didn't really do that. Um, and so they need to do that in the future, bigger cohorts and looking at these different aspects more comprehensively, looking at um, ovarian aging sensitive biomarkers in the plasma, such as what called AMH. So and then there's the whole thing. If you really do want to take this to the extreme and turn this into a translational clinical application, then you got to figure out dosing, treatment duration. Is it more than just drinking a glass of tall orange juice every single day? Is it you got to drink 5 glasses of orange juice every day? I don't know. So there's a lot of work that still has to be done here, but a cool finding nonetheless.
Yeah, though, big up, though, for the level, the detail. Putting omics on a monkey background with, a nutritional study, oh boy. So, yeah, shot in the arm for nutritional studies out there, but, yeah, like you said, that's like we're talking decades. They're still working out that calorie restriction thing with the monkeys
Yeah.
in the age of non-animal models. Watch out. All right, wrapping it up. We're getting kind of back. I guess there's a kind of a theme about fate choice that we're scratching at today, and this is a very in silico story. But I love to highlight these methods as a nature methods story that I think I'm excited to use this. I can't wait to get my single cell data incorporated here. And the theme here really is about, you know, differentiation, the movement of one cell fate to another or multiple downstream cell fates. And that's the key. These lineage diagrams, like, I don't know, I don't want to throw shade. I'm no computer scientist. I can't do better. But sometimes I feel like the lineage diagrams that they spit out, and I'm not going to name any of the program names, sometimes they seem kind of arbitrary. And like if I start at the end and or end at the start, switch around, I get completely different trajectories, which I mean maybe makes sense, but I don't know. It seems like there's a bit of they are left me a bit wanting. Um, and fundamentally, we're talking about like, transcription factors as the major orchestrators of this process. And it's key for understanding not just differentiation, but also reprogramming, right? Getting back to Deepak, his claim to fame. One of the problems there is like, how do you really make sure you have a bona fide product? Also in vivo, like pathologically speaking, understanding maladaptive processes of differentiation like fibrosis, right? So what we need there is a resource to understand the interplay of all these omics architectures, right? The chromatin, the expression. Now we're getting more into the spatial, all comprising these multimodal single cell omics platforms, right? Um, uh, but with those, even so, as I kind of alluded to, the computer science there, the programs, algorithms make it tough to, or they're established to try and reconstruct lineage paradigms because it's a destructive process, right? You can't follow the same cell, right? You have to sacrifice. So yeah, you plug them in to these algorithms, but they can only spit out like relatively small differentiation dendrograms, like little mini trees of a few fate choices, a little snapshot, right? And what we'd like is a trajectory analysis that gives us more coverage and gives us a better signal. So this is a story coming out of multiple leads here, but the lead corresponding here are Rafael Kraman and Ivan Costa, who are from the RWTH Aachen University, which is in Germany, technical institute, the largest public technical institute in Germany. They bang it out there. And they made this program. It's called Flower, PH, with a PH. And they don't even explain the algorithm. Maybe it's some in German. But the HL, at least there, from PHL, for flower, comes from Hodge Laplacian. And the whole thing is decomposition of the Hodge Laplacian for inferring trajectories from flows of cell differentiation. Maybe it comes sweeter off the tongue in German. At least I hope so, for marketing purposes. But yo, who knows about the acronym? But the tech, they benchmark this program, and I don't understand any of that. You got to look at the paper if you're a computer scientist. I understand the output. They benchmark it against all the big names, Slingshot, Monocle, blah blah blah, like 10 of these, and they show that flour does the trick. And here's the key: they use it in this kidney organoid model of data where they have the single cell seq. They got some spatial. They even do like knockout of these three transcription factors based on the trajectory analysis that flour spit out. And all to say that they create these really neat dendrogram lineage trees, but also more comprehensive, like going from mesoderm all the way back to mesoderm and then bam, 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 as opposed to like the dendrograms I get, where it's like a little snapshot with maybe one or two choices in there. This thing was like, it seemed like it was the whole tree, and this is unbiased. So something that you could overlay against a lot of existing data sets and maybe glean some new insight, particularly about like off target cell types in the context of your differentiation trees, because like yeah, these aren't like programmed with the cells that you want or expect. So I don't know. I love a program like this because as you know we get more familiar. Arun, I don't know about you, but you got to get down with our studio nowadays. At least I do because I don't have a bioinformatician in the lab, and it's fun to apply these new modalities to your existing data, and sometimes you can wring out a little extra insight. So I'm looking forward to having another look and dusting off the old data.
Yeah, I mean, it's you don't even have to have any sort of R coding knowledge these days. That's what our GPT friends are for,
Hmm.
right? Even when I was in the Seidman lab, I'll be the first to tell you, um, I was the least bioinformatically inclined of all of the scientists in that really incredible lab. But I knew how to copy and paste code like the best of them. And now I don't even have to do that. I could just ask GPT to write it for me, do the whole shebang.
Hmm.
But yeah, hey man, flower power here. This is good stuff. You didn't name the competition, but they did. They named Monocle. They named the other options out there, and yeah, I agree with you. Like for me, it's less about the comp side, less about the nitty gritty of the code, and really figuring out what the applications are here. Especially, I loved how they incorporated the iPS derived organoid kidney organoid model here, because of course the kidney organoids, as we know, are complex. And we had a recent guest on the show from USC who was talking about these next gen kidney organoids with all the different various cell types that have the structure and the function. So you got to be able to dissect the cells and the organoids using a powerful algorithm like this. So you gotta take the flower power to the next level, I suppose.
I like that. The flower power, the power of the flower algorithm. I'd like to leverage that to vascular diff, right? Because there's, all the different flavors of the rainbow. And it would be interesting to see if there is one common progenitor. I would guess even the flower power would struggle against that problem, but maybe not. We'll have to see. Maybe we'll ask our next guest, Dr. Melero-Martin, but that'll have to wait just a couple minutes. I got a quick message from STEMCELL Technologies. Do you want to keep your laboratory skills up to date? Register for virtual training courses by STEMCELL Technologies to learn how to maintain induced pluripotent stem cells, culture organoids, perform the CFU assay, and more skills. Explore topics at www.stemcell.com/training. All right, everybody. With us today on the show from Boston Children's Hospital, where he is associate professor and endowed chair in cardiac surgery, also an appointment at Harvard Medical School, Juan Melero-Martin, whose lab uses bioengineering principles to understand how vascular networks are formed and the mechanisms by which the vasculature modulates the engraftment and activity of various human stem cells. Current projects in the lab are focused on endothelial progenitor cell biology and therapeutic potential, bioengineering vascular networks, and genetically engineered vasculature for drug discovery. Juan, thank you so much for joining us on the show today.
No, thank you so much for having me. I'm really excited to be here.
Yeah, we're happy to have you on the show. I feel like it's just a matter of time before we had you here. I know you were a listener of the show, so thanks for that. Dalen gave a pretty solid introduction as to what your lab does, but we always like to, ask our guests to give their own introduction and kind of in their own words talk about what they're most excited about in terms of what their lab is working on. So why don't you go ahead?
Yeah, sure. We are essentially we're a vascular biology lab working in the regenerative medicine space. We fundamentally what we're trying to do is we study how the blood vessels are formed, and then we're trying to harness that to use it in regenerative medicine. But the key thing here is that we love blood vessels. I think I fell in love with the vasculature and endothelium a long time ago during my training years, and I still think blood vessels are the coolest thing, that the most important tissue in the body. No offense to other tissues, and definitely my favorite tissue. But now seriously, I think we study here blood vessels because they are so fundamental, right? They're everywhere, and they're essential for the functioning of pretty much every other tissue in the body. And they do fundamental things like delivering oxygen and nutrients, but they're so much more than that. They're signaling centers, and they regulate processes and like inflammation and homeostasis, and or take development, for example, right? Like the blood vessel guide how tissues grow and how even organs form, and or in regenerative medicine, which is our area of expertise. Blood vessels are particularly important because they not also help maintaining tissue, but they also help tissue grow and regenerate, and the interaction of the local stem cells depends on the niches that are formed by this vascular network. So now, it's super exciting area for many of us to study, and very significant. Now the flip side, though, is when blood vessels function fail, there's then usually troubles. You have tissues that suffer, and the loss of vascular function, I guess, is the hallmark of so many diseases, right? And so the focus in our labs, really what keep us exciting is precisely that. We think if we can make blood vessels, we can restore and replace damaged blood vessels. We can make an impact in a lot of areas of medicines. And the way we go about this is we use vascular cells, essentially. I think we're, cells are the protagonists in our lab, and we try to tap into the innate ability of cells to build blood vessels. And so, we can do that with the stem cells, and we certainly love stem cells, along with a range of bioengineering tools, including organized technologies that we're going to talk about today. But I think the overarching goal of our lab is actually simple: is if you can make competent vascular cells, endothelial and mural cells, those are the building block of blood vessels, and you can learn how to assemble them into functional vasculature, you can make a lot of good. And so the lab is just focused on it really boils down to maybe three things, really, like learn how to make ever more competent vascular cells, learn how to engraft those cells in the body, because that's not trivial, and then learn how they assemble into vessels inherently, and then try to tap into that. And the hope is that once we understand how to build these blood vessels from the ground up, then we can use this knowledge to create vascular therapies, or in the engineering space, we can help tissue engineer other more complex tissues and so forth. So that's pretty much who we are.
Yeah, I felt everything you said there. I, like you, love vessels and the vasculature. Arguably my first love, although that might have just been pounded into my head by my postdoctoral mentor, who said, like you, blood vessels aren't just plumbing, boss. In fact, they're everything. And everything, not just in health, but also in disease, as you just described. And yeah, I spent a long time focusing on vascular, also hematopoietic differentiation. And based on what I'd learned about the efficacy, practicality of hematopoietic stem cell transplantation, my thought was always, as a young, naive buck, my thought was always that therapeutic application of vascular cells would be right around the corner, right? But it continues to elude us, despite the tremendous range of conditions it stands to address, as you just alluded to. And part of that reason is that as we've learned, in large part due to your work, is that vascular cells need to be co-transplanted with mural cells, with these support cells, parasites. You could use any number of cell types, but they effectively perform that function. A support cell in the context of co-transplanted with these vascular cells. But you bypassed this need with a pretty surprising approach for me, which was this transfer of mitochondria, which never would have occurred to me. What does this tell us about vascular function, not just in the context of like vasculoplasty, this therapeutic application, but under physiological and or pathological contexts? And is there a way, or what are you thinking about how to exploit this phenomenon therapeutically, maybe to mobilize endogenous ECs even?
Yeah, I think one of the main goals in our life been always, as I said before, engrafting. So, learning how to put the cells back into the body and have them comfortably make blood vessels that are durable and so forth. And as you mentioned, along the way for years, we and others, we have learned that endothelial cells are not particularly good at this. endothelial cells are struggle that initial few days where they're still not part of a really perfused vessel, and then you end up losing the majority of the cells that we try to transplant by themselves. And the way we all have learned to go around is by providing these other supporting cells. We use mesenchymal stromal cells, and other people choose fibroblasts and so on. There's still a range of options that people use. But one question was, how does it work? I mean, how is this other cell supporting endothelium? What is that comes down to? And for a long time, it was thought like maybe, it's the paracrine factors and, survival factors that the mesenchymal lineage is secreting and so forth. And to some extent, there is some of that, but that was not satisfactory enough because that has been people have been trying to, supply those factors, and still endothelial cell engraftment was very poor. So we've been investigating the potential mechanisms of support for a long time. I think a number of years ago, there was one concept that caught my attention that I was really intrigued, which is this notion of cell-to-cell communication via mitochondrial transfer. I was completely oblivious to that, like let's say 7 years ago. And cells exchange mitochondria for way of communicating in all sort of different scenarios I learned. And then, but one in particular was apparently, mesenchymal cells, mesenchymal stromal cells in tissues have been shown in many different tissues to, when you stress those tissues, they transfer mitochondria more actively to the parenchymal cells, the cells, let's say, surrounding, neighboring cells around those mesenchymal. And that's been reported in the lung and in other scenarios, in many different tissues. So we started wondering back then, like, well, maybe mitochondrial transfer is part of how the mesenchymal cell that we're providing to the endotheliums, helps the endothelium. But it was just a little kind of hypothesis there that we didn't know what this is going to pan out, to be honest. But because we have a lot of tools in the lab to track all sort of cellular processes, so we have cells where we can genetically engineer to have fluorescent reporters in the mitochondria and so forth. We just started investigating this question, and we noticed that very soon after implanting the cell, there is stresses, right? When you transplant these cells, there's a lot of stresses for the cell, the nutrient deprivation, oxygen deprivation. The mesenchymal cells start launching these kind of protrusions, like we now know they're tunneling nanotubes, but like crazy. And then they're trying to reach out to every cell around them. And this is 24 hours implanting the cells. And then we'll look at the endothelium that we were co-transplanting with them, and we found that the endothelial cells have received some of these fluorescent mitochondria from the MSCs, right? So we saw the transfer happening. Now, that doesn't mean the transfer was significant in any way, or we didn't know whether that is meaningful at all. But we saw it happening. So then it took us a while, but what we decided to do is we said, well, let's block it. Let's not allow the mesenchymal to transfer. And that took us to a number of months to investigate how can you block that? I mean, but it turned out that the process of this formation of TNT and the motion of mitochondria through those TNT is very well known. To some extent, is regulated by some specific genes, so you can go and block those genes. So we did a number of experiments where we transplant the cells together, but we block the ability of the MSC to form TNTs. Oh, the endothelial cell hated it. The endothelial cell hated the fact that they're not receiving this mitochondria. And then we did something that was actually cool and important. We let the mesenchymal to form the TNT, because through the TNT, not only mitochondria goes, it's a bunch of other things, and microRNAs and protein. there is a lot of things that are transferred, not just mitochondria. So we let the MSCs to form the TNT, but we block the ability of mitochondria to move. You can silence the genes like MIRO1 and things, so the mitochondria cannot just march through those tunnels. Other things can, but no mitochondria. And again, we found that the endothelial cells hate it. So we that was the moment where we realized, wait, okay, so that mitochondrial transfer not only happening, it is actually significant. If you block it, endothelial cells suffer. So we established that this phenomenon of mitochondrial transfer was, in fact, behind this support of endothelial cell engraftment. It was very significant. Now, to your question, okay, what do we do with this? I mean, I guess one of the things that we first do is we try to understand how to encourage the mesenchymal to transfer more or promote this process as a way of improving the co-transplantation. But the leap was something that you mentioned. We at some point we thought, well, how about we transfer mitochondria ourselves to the endothelial cell kind of preemptively, as a way of mimicking the effect that they're having from receiving these mitochondria. This was a leap. I mean, now we understand that it works and how it works, but back then it was a straightforward move. But we did it anyway. We isolated. So what we do is we take a bunch of mesenchymal cells in the lab, sort of like a farm, grow them up, and then harvest mitochondria. And you can do that. That's not overly complicated. And then you take the mitochondria and expose your endothelial cells to them, just putting them in culture. Again, this is not very complicated. The mitochondria get uptaken very natural and easy by endocytosis in a few hours. A lot of the mitochondria goes inside endothelial cells. And then we transplant these endothelial cells. So they're being primed with this mitochondria, the spike of mitochondria. It was very remarkable that we found that even before transplanting, we can already see that endothelial cells have changed in a beneficial ways. Let's say they were producing more ATP, they have an improved mitochondrial respiration overall, but they were also more resistant to stress. That's the key that we were looking for. They were more resistant to apoptosis when you challenge them with stresses. And so that was, I mean, we can get into the mechanism of how it worked, but that was puzzling for a long time, but it was very noticeable though. You can do migration, so you can do any assay, and the cells kind of overperform the ones before transferring the mitochondria. And so now we were transplanting them alone without the explicit direct co-transplantation support of MSCs. And they were surviving they were surviving. The key with endothelial cell is if they survive the period before the incoming vessel from the animal comes and they can connect, you're good after that. But it's that period where is the key. So they were surviving long enough with this extra boost of mitochondria. So these have turned now into what we think it opens up a new way of doing vascular cell therapy with endothelium, which is, priming them preemptively with mitochondria as a way of kind of preparing them to support stresses. And this continues to be something that we're pursuing in the lab. We're trying to see how to translate this and how to make this. There's a lot of questions, obviously, still around how do you make this an actual therapy, but the fundamentals are, it's super exciting. As I say, I mean, I know I'm a mitochondrial guy, and I a vascular biology guy. I learned a lot about mitochondria in the last 5 years or so. And it's a fascinating world. I mean, and the whole mitochondrial transfer, it continues to amaze me, really.
Yeah, I mean, we're always learning new tricks, even as we're moving towards seniority and leading our own labs. But I mean, one of these new tricks that I'm trying to pick up as well are these, vascular organoids, which have really become a very hot topic recently. And we're talking about the importance of co-culture and multiple cell types. And that's kind of inherently what you have in vascular organoids. You have endothelial cells, these mural lineages, and your lab actually really you published this really cool paper in Cell Stem Cell, which I think we highlighted in our roundup a while back, where you had this rapid generation of functional vascular organoids through transcription factor activation of both endothelial and mural lineages. So could you walk us through that paper, in part because I want to just listen into your expertise on vascular organoids. This is something I'm bringing into my own laboratory. Like, how did you arrive at the design of that approach, and talk a little bit about like how these vascular organoids can recapitulate, quote, real microvasculature in their function?
Yeah, so we're being always interested in trying to make the co-differentiation of the vascular cells. That the blood vessels requires endothelium, but requires mural cells, right? Mural cells are the pericyte, the small muscle, the cell that wrapping the endothelium. It's not trivial to have a co-differentiation of those two cell types simultaneously because for the most part, most protocols to differentiate vascular cells rely on specifics, cocktails of growth factors, and so forth. And as it happens, the ones that you need to differentiate mesodermal progenitors into endothelium are incompatible with the ones that you need to differentiate into mural cells. So it's pretty hard to have, at least in a controllable fashion, to have both simultaneously. And so most of the field didn't, I wasn't bothered about that for a while too, because you can differentiate them separately. But there is a point where if you really want to study how blood vessels developed and how everything develops together and differentiate together, you just, it's not trivial. So there are early studies like, embryonic embryo bodies like differentiation, where you can promote the differentiation of endothelium, and then spontaneously the mural lineage kind of shows up in there, but you have very little control over it. So that was something that we always wanted to do. But I think the key here, maybe I turn back to some of our earlier study with the transcription factor, stem cell transcription factors. I think we turned into a stem cell, I guess, like anybody else, because stem cells are really cool and they're really precious. I think it's but before stem cell and iPSL, I have also we work with all sort of, I guess we study with primary cells like anybody else, primary endothelium, and then but I think the consensus about how much translational potential those sources have was very clear that had a lot of limitations. And then we turn to natural progenitors, right? Like we're trying to tap into natural progenitors for the endothelium and for the mural cell lineage. But eventually also figured out that has also limitations. Like the problem is usually the same. Like as we age, and with age and disease, these natural resources of progenitors decline. The abundance and the potency of this. So iPSL came along, and we all super excited. And thanks to Yamanaka and others, we are now in a Nobel Prize week. So kudos to that discovery. I mean, I honestly I was from 2006 to 2012 Nobel Prize. That must be some sort of record.
Yeah. Wow.
So we learned a lot about iPSLs over the last I would say 10 years or so. And now the challenge was differentiation, like everybody else. Then the approach that we took is transcription factors. And so again, I think we thought that if you guide differentiation through the use of transcription factors, you can achieve more control over that differentiation. I tell you this because when we start the differentiation of the vascular cells and endothelial cells, Dalo, you may have experience with this as well. it was a little frustrating. I mean, that you can develop some media's differentiation with the key growth factors and VEGF and all the other growth factors to differentiate your pluripotent cells. And then you get to a point where differentiation is good enough, let's just say 40% or 50% differentiation efficiency, and then we turn to a different clone of iPS cells, a different line of iPS cells, and then it plummets to 5%. It was so at least things had gone much better, but the early years of this was very frustrating. So I have one of the fellows in my lab. It was Kai Wang. He has his own lab now in Peking University. He tried to tackle on this. He was so frustrated about this. One of the things he realized is that the activation of a transcription factor that is called ETB2, which is a master pioneer transcription factor it happens. So mesodermal cells activate this transcription factor to initiate a differentiation to endothelial cells. That was always patchy. It was very difficult to have a synchronized culture of mesodermal progenitor to activate this pioneer transcription factor. And I suppose the reason behind it is because that's not how it happens in nature. When you have some of the cells start to differentiate, they signal the neighbor one not to, and so there is a coordinating process. So with the growth factors, you sometimes run into these kind of difficulties. But if you hack the system, I guess, if you force them to express internally the transcription factors to all of them, you can have very uniform conversion into a lineage that you want. And that's what we did with the ETB2. We also learned with the ETB2 that it's a pioneer factor that is also needed transiently. And that becomes important for later, but it also needed transiently. So it's an amazing story with ETB2s. During development, it comes for 1 or 2 days. It does its thing, goes away, and for the rest of our lives, ETB2 is off. Although this has been disputed too. There's some evidence that it may come back in some angiogenic processing. But for the most part, it's a transcription factor that shows up, do this thing, goes away, off you go to endothelial. So that allows us to develop all sort of systems where the transcription factor is only needed transiently. We don't need to engineer the cell to have it all the time. We just give them a pulse of ETB2, and then cells can differentiate very uniformly. So we were we achieved levels of differentiation and reproducibility with different cell lines, which it was a challenge that was, we essentially transformed our lab, really. That's the way we make endothelial cells now, and virtually most of our projects use this kind of a differentiation protocol with the transcription factor activation. Now, the mural cell was a little less straightforward, I guess, because with ETB2, there was some knowledge out there about it. We didn't know much about transcription factors that guide the mural cell lineage. But what happens is we were involved in a project that was led by George Church here at Harvard. So he was studying these massive transcription factor libraries and the effect that individual transcription factors have on iPS cells and pluripotent cells in differentiation. So he was screening, hundreds and hundreds of human transcription factors and see which one single-handed can differentiate iPS cells into recognizable cell types. So we participated in that study. We were just looking for possible vascular phenotypes. And there was one transcription factor that caught our attention, is this NKX 3.1. That was very intriguing because when you expose iPS cells to NK 3.1, we saw that the cells kind of become something that looked like a stromal fibroblast-like very quickly. That's different than mural periseismal muscle, but it's getting to that. And so from that moment on, we got intrigued, and we just learned about that the key is not to bypass the mesodermal stage. So instead of applying that transcription factor directly into the iPS cells, we make iPS cells we differentiate that into mesodermal progenitor, which everybody knows how to do. This is not complicated. And then we activate NK3.1. And that was a way of generating mutant progenitor cells. Again, it's a story very similar to the ETB2, very efficiently. Like we had conversions in the 90%, 95%. But almost more important than that, very reliable, because you're not giving the cells the chance to people. You're just kind of forcing them to march through that lineage. And so anyway, so this was a little long intro for the STEMCELL paper. But what I want to tell you is, at some point, we were now armed with these two transcription factor strategies to drive both lineages. And then we thought, well, maybe now we can do this simultaneously, because we are not relying on the growth factors. We have switches that we can activate, and we can maybe simultaneously differentiate the mutant and the vascular cell. So we end up forming these, what we refer to as vascular organoids. What we essentially do is we have a genetically engineered lines of iPS cells that are tox-inducible ETB2 transcription factor, and we have other lines that are tox-inducible NK3.1. Then we take them to a mesodermal stage and form aggregates. Right? We keep them in suspension with an orbital checker and form aggregates. An important thing is, at that point, both of the lineages are identical. They're mesodermal progenitors. We haven't yet activated transcription factor, right? This is important because we can mingle them. We can mix them like salt and pepper aggregates. Like, they don't segregate. They are identical cell types. Then we put them in suspension, give them doxycycline. Half of them get ETB2 activated. Half of them get NK3 activated. And they are co-differentiating. Now, what we found is that not only they co-differentiate, you can see by flow cytometry or by other techniques that they are endothelial and they're mural, half of them. You have full control on the proportions, right? And you have full control on the lineage. You can put a reporter of a color in one lineage and the other. You can knock out a gene only in one lineage and not in the other. We couldn't do that before with other systems. But as I say, they're not only differentiated, they're starting to interact. And it was super cool because in really few days, like in two days after activating the transcription factor, we see these kind of primitive networks and cores of endothelium, and then the interstitial mural cells. And then a few days later, we see some of these adjacent mural cells to the endothelium start to differentiate into more muscle-like cells, expressing markers, more mature markers of muscles. And we can even see primitive lumens, very, very tight, because they're not perfused, right? This is a suspension of organoids. But so that was that's the method that we developed. Again, it gives us full control over each of the lineage of the blood vessel component.
Yeah, the function, right? I mean, it's funny because we led with this whole the vessels are everything, and this was such a remarkable recapitulation of that self-organization quality. And I think the key there, as you described, was overriding that paracrine local kind of native impetus there, the cell intrinsic or collective impetus there of cells and development, right? Because that's the thing about blood vessels that's so unique, right? As although they're ubiquitous, they're also highly specific to the organotypic, we'll call them, right? And I remember, as you described, I was having like PTSD as you're describing your postdoc experience there, because I remember the frustration with, yes, absolutely, that was one thing, is that you'd have it all figured out, and then you not even change lines. Like maybe you would just get a deeper passage, right? Like 5 passages are gone, and suddenly the cells would be not only less efficient, but like it would crash, right? And worse for me, was that would you get these perfectly beautiful isolates of endothelial cells, and in 2 days of culture, as soon as they get confluent, they wholesale switch to some kind of mesenchymal cell, right? So like it seemed, and frankly, it was kind of like I kind of gave up. But there were all these glimmers of hope, as there are in science, that pull you along, just keep you in the game, and then dash your hopes once again. And for me, one of those was that, oh, we had this like a subtype of endothelium that we thought, but phenotypically, this is way back in the day before single cell, but we thought it was phenotypically liver sinusoidal EC. So somehow we were recapitulating that differentiation pathway, and those sinusoidal liver putative ECs were more stable, right? So they weren't switching, and that was all our logic. And then we tried to recapitulate, crash, hopes dashed, I'm leaving the field. Point being is that there's this tremendous, I don't want to call it plasticity, but there is plasticity, but also the variability in all the EC subtypes that you get from ES cells. And your paper, you also had this other paper where you really detail that ETV2 override with mod RNA in that case, which was also, I think, another very elegant way of getting that transient burst that you described. But again, getting back to the beginning of my question here, is where we've talked so much about the recapitulating the function, but that organotypic function, right? That angiocrine aspect of endothelial cells. Let's talk about that. So with your ETV2, you've kind of overrided. Now, have you? Because Shaheen will tell you that ETV2 also it's a gateway transcription factor that makes these ECs that can do whatever you want. You put them in whatever niche, and they assume that angiocrine profile. If you deploy these ECs that you've been working with, do you see some of that kind of organotypic function? Are you looking down those paths? Talk a little bit about that angiocrine.
Angiocrine well, listen, that is one of the. I mean, there's a lot of outstanding questions still, but it definitely is one of the directions that a lot of us are looking at is the tissue specificity. How to make, I mean, and there is many degrees of tissue specificity. So the endothelium is a very heterogeneous lineage. I think it's not a monolith. If you work on it, you know that. Like you can start with basics like arterial, venous, lymphatics, or you can go deep down into every tissue. And then these days we have these beautiful single cell atlas analysis of tissues, and you can see how heterogeneous the endothelium is in every tissue. So I think one of the, again, directions that many of us want to continue to work on is precisely on how to make our endothelium acquire that tissue specificity. Now, I think it's important to know one thing, though. There's definitely tissue specificity of the endothelium. A different thing is to understand it. Like, okay, what is what to make to understand the meaning of those differences is something that we don't collectively have a good hand on. In other words, we can appreciate and see the difference. We can see all the transcriptional profiles and genes that are on and off and all that, but that doesn't mean we know how to make sense of what those do. So because of that, there is still some of us, and I was in that camp for a while, that thinks like maybe the key is not so much to learn, because it's going to be complicated to learn how to make liver specific or brain specific disease, but make something more like epigenetically naive endothelium or plastic. And when you deploy these cells, and I'm just thinking about transplanting now, but when you transplant these cells into a different organ, because of that plasticity, the cell will acquire the. There are people who have shown, Shaheen Rafi being one of them, that has shown the validity of this concept is moving in the field, but for the most part, it remains a little theoretical. Mm-hmm. So we have not proved any of that. I mean, for the most part, we implant our endothelial cells either subcutaneously or intramuscularly. And yeah, we can try to see whether but we've been more focused on whether we can create arterial and venous heterogeneity. That's the level we are right now. But there is no question about that. Another frontier is that level of capillary specificity. I think that the learning, the transcription factors that may regulate this, it can also be an avenue of opportunities there for young researchers to investigate how to acquire, because the growth factors, I think mimicking and reproducing the complexity of the tissues where that specifications happens in vivo and during development is more complex than we may want to acknowledge. But yeah, I agree with you 100%. That's a direction that not just our lab, many other labs are looking at. And there is two camps, so we're exploring two kind of things: learning hardcore how to differentiate them, or learning how to make them plastic enough that they will differentiate.
Yeah, there's just so much to dissect, and I feel like we could go on for another 2 hours just talking about the intricacies of not just the model systems, but the mechanisms of how to make these new model systems like vascular organoids even better. I mean, we've talked to so many folks here on the show who are working on iterations of technologies like vascular organoids and hybridizing them to make assembloids. And I just feel like there's an infinite rabbit hole that we can go down. And perhaps we can have you again on the show in the future. But I wanted to wrap things up with a question about where you are. Obviously, you're at Boston Children's, affiliated with the Harvard Medical School, Harvard Stem Cell Institute, which is one of the, as we know, one of the world leaders when it comes to all things stem cell biology. It's an incredible ecosystem, and one I've been able to experience at different stages of my own career. Like 15 years ago, I was an HSCI summer intern at the Brigham. Actually, no, it was at Beth Israel. I mean, it's been so long. And then I did my postdoc actually at HMS Genetics as well. So it's really just an amazing place for when it comes to stem cell biology. And what is it about that ecosystem? The people, resources, collaboration, culture, the trainees that really makes it such a compelling home for vascular biology?
Yeah, I think, there is an unusual concentration of talent, but also there is I think there's a culture of innovation that it take us, a long time back. I think I've been here like about 25 years now. Came for my postdoctoral training to join the vascular biology department at Children's. And I was led by Dr. Judah Folkman the late Judah Folkman at the time. And so I was fortunate enough to see the Dr. Folkman and to interact with Dr. Folkman for the last, 2-3 years of his life. And the culture that I was creating in this, I was super fortunate to be in that department. And that's why I fell in love with, I'm a chemical engineer by training. I didn't have a lot of vascular biology knowledge before I came to Boston. Don't mind admitting that. During my PhD, I was trained in progenitors and cells and things, but the love for the endothelium was when I landed here. So such a magnetic personality, Dr. Folkman, this transform. And then, so there is, he's been promoting vascular biology here at Children's for, 50 years before I came. Huge discoveries happen here. There is a culture of innovation that is contagious. You're doing your postdoc for the first time, and in the other aisle is Michael Klasburn, who discovered base EGF, or Judah Folkman, who discovered. It's just an amazing place to be. A different thing is once you start turning into being faculty. Well, it's not different, but it's so postdoc is awesome to be exposed to all this, right? This culture. Starting your own lab here at Boston is, you need to understand who you are, though. I think I'm a person who likes to be surrounded by people that are more smarter than me. Let's just say that, okay? And I get inspired by that. But it comes with challenges, you know. You have very little visibility. So there's so many big names around here, so many things going on that when you're starting, so you gotta be careful. You gotta understand who you are. You gotta be strong and just put your head down, work, and hopefully have good mentors and have good and you can get all this in Boston, but you know it can be tricky. Let's just say that it can be tricky. I've seen people who fell behind this if you're not mentally strong.
Yeah, talk about a big pond. And you mentioned Judah Folkman. I think
Yeah.
one of the greatest things about him, I joined Shaheen's lab right before Dr. Folkman died. And Shaheen, you know, who's can be a polarizing personality, really admired Judah Folkman. And I think that was common. He was such a unifying force. And I think part of the gift that he had as at a place like Harvard, or I mean, you know, at these most, you know, esteemed institutions where he was operating, he made space for the people behind him, and I think that was really appreciated. And while it is, you know, I guess everyone's always looking over their shoulder. I don't envy you guys who are operating over there.
Yeah.
But you got to recognize that as tough as it may be, as you just described, for the trainees there to be there, and they really are the engine. I'm sure you'll agree, because they're so bright, right? They have that raw intelligence and and the ambition and the energy, and it's a tough road to hoe for the PIs, I'm sure. But wow, the talent that you have at your disposal and the responsibility to that, it's you know.
And we've been privileged to, so I feel I've been privileged in my career that you know, I came to this field at a time where it's a very multidisciplinary. So you have engineers and surgeons and biologists and all, you know, maybe 20 years ago, that was not that common. So we are now, I think it's a golden era of research. I know there is a lot of challenges, and I know there is a lot of preoccupation of you know, political and non-political things that are happening. But what a moment to be in science, though.
Oh man, we talk about it all the time. We
Right.
love our lives to be able to you know, just live in this playground.
And then for the cell therapy point of view, if that's your theme, I think we're on the verge of a, you know, I feel it, of a revolution of different cell therapy products and coming from iPSOs and so it is super exciting. I try to keep my trainees not to lose sight of it. They get worried about the current situations, and obviously they get worried about the future of academia and the ever increasing dilemma of whether to continue in academia or not and go to industry. And say it doesn't really matter when you're doing research. If your passion is research, I mean, it does matter in a sense that you have to understand the differences, but if your passion is different, there's so many options these days. You can be a researcher if you love teaching, you can be in a campus and teaching and doing research. You can be in a hospital like I am, in a research institute, or you can be in Vertex or doing research. I mean, in a company doing state-of-the-art research too. So I don't know. I think it's a fascinating time. And let me just go back to Dr. Folkman, because Dr. Folkman was kind of a character in the sense that he didn't, you know, the way he treats people, it just makes such an impact on people that I was I was doing my postdoc there. I didn't directly interact with Dr. Folkman too much. But he's the kind of person who will see, he light up if he see you're working late in the evening, right? And then he just light up. And then I remember he sees me one day and he'd say, "Hey, oh, why are you working out?" And then I explained to her I was working at the time on EPCs and endothelial progenitors and stuff like that. And I let her reflect on it. At the time, I didn't realize what he was doing, but he made me feel like the things that I were investigating are the most important thing in the world at the time. Like, and he has that ability of making you feel like that. And then a few months passed by, and then he's already in his 70s at the time. Meet me in another meeting. Remember exactly what we talked about it. It's just crazy to me. That was crazy to me. And I say, well, that's what he is who he is, I think. And I obviously I cannot be like Dr. Fuckman, but I try to learn from him when I try to inspire my trainees and just try to leave them with a everlasting kind of impact. That was amazing.
Yeah, well, it's no coincidence that here so many years after his loss, where it's like a little mini homage, and it's merited. He really was a great scientist. All right, well, moving to the post here, talking about you, not Dr. Folkman, Juan. So let's finish with just a little bit of insight into your life away from the bench, maybe, with some of these science peripheral questions. First, what's the best piece of advice that you've ever been given, either professional or not?
Yeah. I mean, I think I've been fortunate to have great mentors, I'd say. So one of the advice that cannot always stay with me is, it's a little strange, but is something like learn to be comfortable with failure. And I think that whether we like it or not is it's a big part of our profession. I think we're investigating cutting edge things. So experiments will logically fail, or hypotheses will be wrong, and you get papers and grants rejecting all that. And to be comfortable with that is not natural. It is not natural. Almost no one, not trainee that I get comes already trained for that. And so I put a lot of effort in getting my trainee to understand this, to channel kind of negative feedback into something positive, rather than get frustrated. I think that mindset have served me well in my career, and it's not easy though, but that's an advice that I always remember.
Yeah, isn't that the truth as a scientist? I mean, you can't be embarrassed to be wrong. The number of times I've put out an idea with my trainees, like, oh, maybe it's this, and then like within hours even be proven wrong. But the real failure would be to run out of ideas, right, Juan? I mean, it's and that failure is our friend. Finally, we got a little run of fill in the blanks here.
Okay.
When I am not conducting research, I am.
Well, if I'm home, I'm gonna tell you I have two passions. One is watching soccer. I'm really I am a huge Real Madrid fan. So I follow European soccer and since I'm a kid, so never miss a match if I can. And the other passion is cooking. I'm most likely gonna be on my kitchen cooking something for my family. And the thing with cooking, I think it reminds me to be in the lab in a way. And these days in the lab, my trainees do the work, and I barely do things with my hands anymore. There is something about doing things with your hands, and I think the kitchen and cooking reminds me to that. But anyway, I love cooking. I cook very often. I love experimenting with different cuisines and everything. So I'll say that probably either of those things.
I'm with you. I love to cook. I have a garden and I have the lab. That's my triangle. And the lab is like my winter garden. The garden you can go into every day and see what's growing. It's the best. If I could have one superpower, it would be.
Ooh, one superpower. Okay. Maybe teleportation. I have a pretty long commute. It takes me an hour sometimes from home to work and every day. So it'd be amazing to just teletransportation or whatever it's called, and or go back to Spain and then get back instantly. That would be a superpower.
That is it. A traveler. You're like my
Yeah.
son. He hates to fly, and he wants to invent teleportation. I told him to go with it. He'll bring the idea to you. You guys can share the patent. Finally, I can't start the day without
It's coffee.
Of
I mean,
course.
I'm not a human being unless I have coffee in the morning. I think also one of the first things that I do every morning is to actually listen one of these short daily news podcasts. I do this a lot. So definitely part of my morning routine, some news and coffee. Yeah.
Get informed, get caffeinated, get out there. Juan, I'm happy that you joined us. This was such a fun talk, and for me to talk about endothelium with someone who's on the inside track like you is a great joy. And what I've learned is that there's so much left for us to discover. So thanks for joining us, and we'll have you back on the show soon to continue the discussion.
Thank you so much for having me. This was fun.
All right, everybody, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCellPodcast.com, or via email at info@stemcellpodcast.com with feedback or to suggest guests.